Okay, so here I have a DNA replication bubble drawn, and this is pretty much just an opening in the DNA, which is unwound DNA, which allows for replication to occur. So first, I'm going to go ahead and start by placing helicase where it goes. And it goes towards the opening of the replication fork, and it heads out of the replication bubble. Helicase is the only enzyme involved in DNA replication that I'm going to be drawing today, and it pretty much just unwinds the double helix from the DNA by breaking hydrogen bonds between the nucleotides. Alright, so next I'm going to go ahead and I'm going to place the primers on the leading strand. So the leading strand is going to be 3 to 5, and the primer is going to be placed right next to the origin of replication. This will be the same on both sides. So the leading strand is synthesized continuously, so that means it's moving towards the replication fork. Primase lays the prior DNA poly 3 adds nucleotides in a smooth and uninterrupted chain, and then DNA poly replaces the primer and ligase seals the whole thing, so that's pretty much the leading strand. Now I'm going to go ahead and place the primers for the lagging strand. So there's a space between the origin of replication and the primers. There's going to be multiple primers on the lagging strand, and these are called Okazaki fragments. This is because the lagging strand is synthesized discontinuously in short segments. So to label them, one is going to be closest to the origin point. So it's going to go 1, 2, 3, and then on same on this side, 1, 2, 3. And these are actually going to be opposite primes. So right here is going to be 5 and 3 for each of these primers. So I'm going to go ahead and label the lagging strand. This will be the leading. Okay. So pretty much on the lagging strand, there's fragments because it runs in opposite directions of the replication fork. So primase lays down multiple DNA primers, and then DNA poly 3 builds each fragment in the 5 to 3 direction. And DNA poly replaces the primers and ligase seals it all up. So that means DNA synthesis is going to move from 3 to 5. So I'm just going to draw the arrows in. So everything up here is moving in this direction. Everything down here is moving in this direction. And that is my DNA replication video.